All right, well, welcome everyone. So we are going to begin the process of looking at some physiology as it relates to our circulatory system. So we're going to look at blood vessels and specifically look at how they function in terms of circulating blood, maintaining blood pressure, things like that, exchange of gases and nutrients at the capillary. We'll look at those types of things, right? So one thing I want to point out first is just the kind of basic overview of what's happening in our different blood vessels, right? So if you look at this graph, what you'll see is along this axis here, we've got different types of vessels, right? So elastic arteries, so think aorta, immediate branches of the aorta, muscular arteries, so think like brachial artery, femoral artery, things like that, arterioles, capillaries, venules, veins, and then your superior and inferior vena cava, right? And so what you can see is that as you change the cross-sectional area, right? So essentially the cross-sectional of one vessel is the diameter of that blood vessel, right? The cross-sectional area of a vessel is the luminal diameter. So as you increase the cross-sectional area of a vessel or of a vessel type, right, you can see how there's a corresponding change in the flow of blood through those vessels, right? And what you see is that it's an inverse relationship, right? So as we increase cross-sectional area, we begin to have decreases in blood flow velocity, right? Well, we know that cross-sectional area is highest actually in some of our smallest vessels. So our small arterioles, capillaries, and our venules, right? And through those blood vessels, you have a slower velocity, right? A slower movement of blood, if you will, right? So again, in this case, it's measured in centimeters per second, right? And so part of this you can think of it having to do with just the nature of the vessels themselves, right? So arteries have more smooth muscle in their tunica media compared to veins, which have proportionally less smooth muscle, things like that. The arteries also have the heart behind them, right? So when the heart contracts, you get that big flow, that big movement of blood, whereas you don't necessarily get that with the veins, right? And then in the capillaries, it's slow because you need that slow movement to allow for the exchange of nutrients, gases, things like that, right? So just consider that as we begin talking about movement of blood through our vessels, blood pressure, and things like that. Okay, so one thing that we'll look at is this process of capillary exchange, right? Capillary exchange. And so this is essentially when we have our things in the blood, right? So think oxygen, think carbon dioxide, think all of our other nutrients that we have in the plasma portion of our blood. This is where we're going to begin to exchange that between the blood itself and our various tissues, right? So wherever those tissues are, in the brain, in the muscles, in the liver, we're going to have the exchange of those types of nutrients, right? And so one way that we have the exchange of material is through diffusion, right? So we're called diffusion. This is a passive process, right? So think passive transport, moving substances from a high concentration to a low concentration, right? So from the blood to interstitial fluid, or from and then to the tissue, or from the tissue to the interstitial fluid into the blood, right? So for different molecules, for different gases, the movement or the direction of the diffusion is going to vary, right? So if you think about oxygen, right, oxygen is going to diffuse one direction from the blood ultimately into the tissue, and carbon dioxide is going to diffuse in the exact opposite direction from the tissue ultimately into the blood. And then when we get to the lungs, the exact opposite is going to happen, right? Oxygen is going to go into the blood, and carbon dioxide is going to go out of the blood into the lungs, and we'll breathe that out, right? Other things, so larger solutes, so think maybe proteins or amino acids or something like that. These may have to move, or fatty acids, these may have to move through gaps in our capillaries, right? So when we talked about anatomy of different types of capillaries, there's fenestrated capillaries, there's sinusoidal capillaries, there's the continuous capillaries. And so the openings in those capillaries in the basement membrane and in the endothelial cells, that's also going to influence the movement of solutes into and out of the capillaries, right? Now, you've also got another type of movement, still at the capillary level when you have the exchange of nutrients, but this is called vesicular transport, right? Vesicular transport. And so, hence the name, this is using vesicles, right? So recall, maybe from your AMP1 discussions, that vesicles are these kind of membrane-bound structures that we find inside the cell that result or cause the movement of material either from the inside of the cell to the outside or from the outside of the cell to the inside, right? So if it was moving things out of the cell, we call that exocytosis, right? So exo meaning out, external. And if it was moving things into the cell, we called that endocytosis, right? Endocytosis. So again, a lot of it is through diffusion, but again, you have this process called vesicular transport, right? Now, one type of endocytosis is this thing called pinocytosis, and think of pinocytosis as like cell drinking, right? The cell is endocytizing fluid into itself. It's bringing things into the cell. Into the cell, right? So larger molecules are going to be moved this way. Maybe things like proteins are going to move into the cell via this vesicular transport type of process, right? We then have this concept called bulk flow, right? So this is the mass movement of fluid as a result of different pressures that we're going to see in just a moment. So we're going to talk about a couple of different pressures, one called a osmotic pressure, one called a hydrostatic pressure, and the interaction between these two pressures and the extent to which we have one or the other, that's going to result in this bulk flow of material from the inside of the blood vessel to the outside and from the outside in, right? And it's going to be built or it's going to be dependent on pressure differences, right? So depending on where the highest pressure is and where the lower pressure is, we're going to either get filtration, okay? So we're getting filtration in which things move out of the blood, so blood to the interstitial fluid to the tissue, or we're going to have reabsorption, where things go from the tissue to the interstitial fluid, ultimately back into the blood, right? So again, we'll talk about a couple of different types of pressures, right? So the first type of pressure is called hydrostatic pressure, right? So if you look at the word hydrostatic, right, just hydrostatic, hydro is referring to fluid, right? So think hydro, think water, think fluid. And then static is meaning kind of the state of not moving, right? So just the force of the fluid, the force of the water that's being exerted, that's hydrostatic pressure, right? And you have two different hydrostatic pressures because you have fluid in two main areas. You've got the fluid in the blood, right, in the blood vessel, which is because of the blood, and then you have the hydrostatic pressure that's in the interstitial fluid, right? And that is the pressure of the fluid that's in between the blood vessel and the cells, right? So interstitial is that that space in between the tissues and cells and the blood vessel, right? So it's a type of extracellular fluid, right? And so depending on which of these pressures is greater, right, which one is larger, then you're going to get a net hydrostatic pressure in one direction or the other, right? So if we're having filtration, if we're having things move out of the blood vessel, then that indicates that the blood hydrostatic pressure needs to be larger than or greater than the interstitial fluid hydrostatic pressure, right? And again, it's a result of the amount of fluid that's being the fluid itself is putting the pressure on the fluid around it and on the space to result in the movement of materials. And typically the blood hydrostatic pressure is going to be larger. The interstitial fluid hydrostatic pressure is going to be a little bit smaller. All right. Now we also have one called, it kind of has two different names. You may see it called osmotic pressure, just osmotic pressure. You may see it called colloid osmotic pressure. Sometimes you may see it called oncotic pressure. right? But they're all referring to the same thing, right? And so this osmotic pressure, this is due, when you think of this as being the result of proteins in the blood, right? So there's a protein in the blood, and that protein is acting, it's exerting an osmotic pressure. So think of osmosis that you talked about in maybe AMP1. Osmotic pressure or oncotic pressure, think of it as holding and pulling fluid, right? So it's wanting to keep fluid in the space where you have the proteins, right? So blood colloid osmotic pressure is the pressure exerted by the proteins in the blood, right? So think albumins, right? In this case, if it's a protein in the blood and this pressure due to these proteins is trying to keep fluid in that same location, then this blood colloid osmotic pressure is opposing filtration, right? It's resulting in a net movement of things into the blood vessel, right? Interstitial fluid osmotic pressure, or colloid osmotic pressure, is the exact opposite. So this is the pressure of proteins in the interstitial fluid that's trying to keep and draw water into the interstitial space, right? Now, usually this is very low, if there's a pressure at all, because there are very few proteins in the interstitial fluid, right? So again, typically the blood colloid osmotic pressure is going to be greater than the interstitial fluid colloid osmotic pressure, right? So two main pressures, hydrostatic and this colloid osmotic, and then for each you have the pressure in the blood and you have the pressure in the interstitial space, right? So what you can then do is you can take all of the pressures that promote filtration and then subtract all of the pressures that oppose filtration, and what you get is something called your net filtration pressure, right? So it's the difference between the net hydrostatic and the net osmotic, right? And so again, you're essentially looking at all the pressures that could potentially result in things moving out of the blood, and look at all the pressures that are promoting things moving into the blood, right? If you want the movement of things out of the blood, so you want filtration, then you need a positive net filtration pressure. If you want things moving back into the blood vessel, so reabsorption, then you'll need a negative net filtration pressure, right? And so you get both of these, positive and negative. So you get one promoting filtration and one promoting reabsorption, but at different ends of the capillary, right? So on one end, you'll get the pressure that is promoting filtration on the arterial end, right? So on the side closest to the arterial end, and I'll show you an image in a moment. And then on the venous end, so closer to the venule, you have the pressure that's promoting reabsorption, right? So you can think about which of those pressures, again, blood hydrostatic pressure, interstitial fluid hydrostatic pressure, blood osmotic, or interstitial fluid osmotic, which of those and what would they need to be in order to result in a positive net filtration pressure or a negative net filtration pressure, right? So what you can do is you can essentially just plug in numbers, right? And if you look in a textbook, right, oftentimes they will give you they may give you example figures or example numbers that you can plug in to get your net filtration pressure. So it may be like 32 millimeters of mercury or something for your blood hydrostatic pressure, and maybe it's 25 for your interstitial fluid hydrostatic pressure, and then for your osmotic, maybe it's 10 on one and it's 12 on the other, right? The numbers you can kind of look up, but plug those in, do the simple math, and then see what you get, right? Are you getting a net filtration pressure that's positive, or are you getting a net filtration pressure that is negative, right? So what you can look at is an image kind of like this, right? So there's a lot going on here, but it's simply what we just talked about, right? So all we've done is we've added in figures. So again, here's our colloid osmotic pressure here. So net colloid osmotic pressure. Here's net hydrostatic pressure, and you get net filtration pressure. Notice here that it's a positive number, right? Positive 14. You have the same two sets of parameters here. So net hydrostatic, net call it osmotic. But notice here our net filtration pressure is negative, right? So a negative net filtration pressure indicates that fluid is moving into the blood vessel. A positive net filtration pressure is indicating things are moving into, or I'm sorry, things are moving out of the blood vessel, excuse me, right? So this is filtration on the arterial end. This is reabsorption on the venous end, right? So obviously as you have alterations, as you have changes in the pressures, you're going to have changes in the net filtration pressure as well, right? Now some of the fluid goes out during filtration, right? There's some amount of fluid that goes out. There is some amount of fluid that comes back in, right? But it turns out that not all of the fluid goes back into the blood vessel, and it accumulates or it stays in that interstitial space, right? And so this is where the lymphatic system comes into play, right? So the role of the lymphatic system, right? So we'll have separate discussions and lectures and talks about the lymphatic system. It kind of has a relationship with the immune system, but it also has a close relationship with our cardiovascular system, right, in our circulation. And so if the fluid that goes out during this capillary exchange, it goes out into the interstitial space, some of it comes back in, but not all of it, the result is that some of it will need to be picked up by our lymphatic capillaries, right? So something like 3 liters per day of fluid filters out of the capillaries, and any of that excess gets picked up by the lymphatic system. So again, maybe like 15% or so circulates through the lymph vessels, so capillaries, vessels, ducts, and trunks, and then ultimately it gets returned back to the circulatory system. Right, so lymphatic fluid comes from the blood, ultimately comes from the circulatory system, and it's ultimately returned to the circulatory system. Right, so consider some of these questions, and we will pick up in our next part talking about local blood flow.